Looking at this fossilized skull, the heavy brow ridge and elongated facial structure immediately distinguish it from our own. These morphological differences define our closest extinct relative, the Neanderthal, and highlight a persistent biological mystery. What exact evolutionary mechanism allowed our specific lineage to survive while theirs perished? The answer is hidden in a biological paradox. We share roughly 99% of our DNA with Wevugreid apes like chimpanzees, but that remaining 1% still leaves a massive 35 million base pair gap. Somewhere inside those 35 million genetic differences is the precise sequence of mutations that constructed the anatomically modern human. We cannot close that gap by studying chimpanzees or bonobos. They branched off far too early in our evolutionary history. To isolate the exact genetic code responsible for our modern traits, we have to look at the hominins who shared our world until 40,000 years ago. But if we plunge beneath the surface of a 40,000-year-old fossil, without a living cell's repair mechanisms, we are left with shattered, decaying double helix fragments buried deep within the fossilized cellular matrix. Early paleogenomics researchers, led by scientists like Svante Pääbo, recognized that these fragile fragments were the only remaining window into that 35 million base pair gap. They began extracting and sequencing this degraded material. By successfully reconstructing the Neanderthal genome, science moves from interpreting the shape of dead bones to reading the exact biochemical recipe of our species' survival. Extracting that sequence begins with an immense physical challenge. These specimens have been degrading in damp, layered dirt environments for tens of thousands of years. Because of this exposure, over 99% of the DNA extracted from an ancient bone belongs to the soil microbes and bacteria that colonize the remains. Filtering out that microbial noise required a technological leap. Instead of trying to amplify specific, targeted DNA fragments, researchers moved to high-throughput sequencing. These machines blindly sequence every single fragment extracted from the sample, yielding millions of genetic reads. Once the microbial DNA is isolated and removed, researchers face an even harder problem: modern human contamination. Because our DNA is so similar to a Neanderthal's, geneticists must mathematically prove that the primate sequences they are reading are not simply from the shed skin cells of the archaeologists who handled the bone. This sequence alignment demonstrates how they prove authenticity. Over millennia, the cytosine bases, the Cs in the DNA sequence, spontaneously decay into uracil, which the sequencing machines read as thymine, or T. By tracking these predictable chemical errors at the ends of the DNA fragments, researchers can verify the age of the sequence. They also employ strict biological fail-safes. By sequencing bones known to belong to female Neanderthals, any trace of a modern male Y chromosome is immediately flagged as contamination. They also cross-reference specific mitochondrial DNA sites where all modern humans match, rejecting any fragments that carry the modern signature. Overcoming these chemical and biological hurdles was the prerequisite for turning heavily contaminated dirt into a pristine, mathematically verifiable ancient genome. With a clean Neanderthal genome, geneticists could finally test our evolutionary timeline. Based on early genetic data, the lineages of modern humans and Neanderthals diverged completely roughly 550,000 to 800,000 years ago. This supported the traditional out-of-Africa hypothesis. The prevailing assumption was that anatomically modern humans left the continent and strictly replaced archaic hominins across the globe, without any interbreeding. To verify this, geneticists use a pairwise allele matching test. This phylogenetic tree establishes the baseline expectation. If Neanderthals split from our common ancestor long before modern humans separated into regional groups, the Neanderthal genome should have an equal genetic distance to a modern African genome as it does to a modern non-African genome. But the data revealed an anomaly. The Neanderthal sequence consistently matched non-African populations, Europeans, Asians, and indigenous populations of the Americas, statistically more than it matched African populations. This data birthed the leaky replacement model. It proved that as early modern humans migrated out of Africa, they encountered and interbred with Neanderthals in the Middle East and Eurasia, picking up new genetic material before dispersing across the rest of the world. The ultimate statistical consequence of that admixture is visible today. Modern non-African populations carry a persistent 1 to 4% Neanderthal genetic contribution in their DNA. The genomic data conclusively proves our ancestors didn't simply conquer and replace other hominins. We absorbed them, entangling their evolutionary history with our own. Transitioning away from population statistics allows us to look at the active protein-coding genes. This reveals exactly which physical traits Neanderthals possessed and which ones they passed down to us. Looking at this representation of the MC1R gene locus, researchers found a unique mutation in Neanderthals. This gene controls melanin production. The Neanderthal mutation made the resulting protein less efficient, triggering a biochemical pathway that creates a phenotype of pale skin and red hair. This trait provided a distinct advantage in high-latitude environments. Pale skin allows sunlight to easily penetrate the outermost tissue, optimizing vitamin D synthesis in cold, low UV regions. Researchers also analyzed the FOXP2 gene, which dictates the complex oral and facial motor control required for speech. They found that humans and Neanderthals share the exact same derived FOXP2 variant. This strongly suggests that the genetic foundations for language predate our evolutionary split. Beyond physical appearance and speech, Neanderthal intergression left a lasting health legacy. The genetic variants we inherited from them include alleles that accelerate blood clotting, useful for surviving hunting injuries, specific immune system responses, and even genes associated with depression triggered by circadian rhythm disruptions. Neanderthal DNA is not a dead fossil record. It is active biological code still regulating the immunity, appearance, and health of billions of people today. If we share so much genetic material and evolutionary history with Neanderthals, how do we isolate the mutations that make us uniquely modern? Consider this representation of a population grid. To find our unique biology, geneticists look for an event called a hard sweep. This occurs when a single, highly advantageous mutation appears in an ancestral population. Because the trait is so crucial for survival, it rapidly proliferates across generations. As it takes over, it completely wipes out all the surrounding ancestral variation, leaving a highly uniform genetic sequence across the entire surviving population. Researchers search our modern genome for these long, uniform regions that are completely distinct from the Neanderthal sequence. When they find a sequence where every living human matches and the Neanderthal differs, they have isolated a uniquely modern adaptation. One prime example of a hard sweep is located at a gene locus associated with cleidocranial dysplasia, or CCD. Notice how this wireframe highlights the physical changes. When this specific gene is inactivated by mutation in modern humans, the resulting condition mimics distinct Neanderthal skeletal traits. The modern human develops a bell-shaped rib cage and a noticeable protrusion of the forehead, known as frontal bossing. By mathematically subtracting the Neanderthal sequence from our own, science exposes the precise, recent mutations that constructed the anatomically modern human. For centuries, static museum galleries like this one constrained our understanding of human origins. The narrative was built entirely on the physical shape of isolated, fragmented bones. Paleogenomics replaced these estimates with a high-resolution, quantitative map of our genetic past. We no longer rely solely on the shape of isolated bone fragments, but can now read the exact instructions that built our ancestors. This data redefines the Neanderthal legacy. As active contributors to our biology, they supplied the vital adaptations that allowed migrating humans to survive high latitudes and foreign pathogens. Sequencing the Neanderthal genome ultimately provided the definitive baseline needed to understand our own species' rapid ascent. Isolating our unique genetic innovations explains how Homo sapiens survived, while all other hominin branches failed. The Neanderthals did not entirely vanish. Fragments of their ancient code secured our survival, leaving us as the sole custodians of an entangled genetic legacy.